Welcome again to the practical session of the Dentin lecture. I hope you have listened already to the Dentin lecture so you can keep up to the information I'm going to discuss today in the lab. I'm Dr. Hiba Zer. Let's begin. We will take a look at all the histological features and components we talked about in the theoretical lecture. So we are going to discuss and see the dentinal tubules, the odontoblastic processes inside of them and other components of the dentinal tubules, the mantle dentin, the interglobular dentin, Tomes layer, hyaline layer, the circupulpal dentin, the predentin, the primary and secondary curvatures of odontoblastic tubules, and the lines related to them. We call them in the lecture Schrager and Owen lines. The incremental lines of dentin, which we call them in the lecture von Ebner's line and Anderson lines, the neonatal line, which is one kind of Anderson lines. Also, we will talk about and see some histological slides that discuss the physiological and pathological dentin formation, like secondary, tertiary, and sclerotic dentin, and some phenomena that can be seen in the dentin, like the tracts of fish. So, as we have discussed in the lecture, dentin forms the bulk of the tooth. It's composed of large number of parallel tubules that lie in mineralized collagen matrix. They have a path from the dentino-enamel junction into the pulp, which this path takes and is shape that we called the primary curvature of odontoblastic tubules. The dentin has two types: the one that is inside the tubules, the one that is between the tubules. The one inside the tubules is 10% more highly mineralized than the one between the tubules. The one inside the tubules is called the intratubular dentin or the peritubular dentin, and it can be seen only in partial demineralized or ground sections, as you can see here. While in demineralized section, we can see a space in the place where the peritubular dentin was. The dots that we can see inside the tubules are the odontoblastic processes of the odontoblasts. B here refers to the intertubular dentin. Intertubular dentin. Under electron microscope, as we can see, the dentin between the tubules is the intertubular dentin. The dentin inside the tubules is the peritubular dentin. These are the tubules. In a higher and higher magnification, we can see the tubule with the process inside, the collagen fibrils. There is high collagen percentage in the intertubular dentin, as you can see here, in comparison to the peritubular dentin. This is a section taken from the crown, as the surface area is smaller from the enamel side to the pulp side. If we take a cross section near the enamel, we will find lower percentage of dentinal tubules, while a higher percentage of dentinal tubules is seen when you take a cross section near the pulp, which means the permeability and the sensitivity of the dentin is higher when we go toward the pulpal tissue. A high percentage of collagen fibrils can be seen in the dentin, as you can see in this picture. It stains these fibers. As we discussed in the lecture, the main type of collagen that is seen in dentin is collagen type 1. 90% of the collagen found in the dentin is collagen type 1. So dentinal tubules branch at the terminals, whether the terminals are at the end or the junction with enamel or at the junction with the cementum. So as we can see here, this is a dentino enamel junction with the scalloped shape we talked about. These are the dentinal tubules. They are branching at the end, making a tree-like appearance. Another section here shows the dentinal tubules in a higher magnification. They are so obvious here and easy to see. The branching also is very clear. This is the dentino enamel junction with a little bit of scalloping, which means this area has a moderate occlusal forces. Here is the enamel. And do you remember these structures that goes out from the dentine into the enamel and they don't homogenize with the enamel prisms? We called them last lecture the spindles. Look how there is more spaces between the tubules near the enamel, as I just told you that if we take a cross section here, we'll have a low percentage of dentinal tubules. We have more intertubular dentine in this area. The dentinal tubules also have some lateral branches, which means they branch in the pathway from the enamel into the pulp. In the way they communicate with each other through lateral branching. That can be seen here. Also, the dentinal tubules don't take a straight pathway from the enamel to the pulp. They actually go sometimes up and down in that pathway in what we call secondary curvatures. A higher magnification that shows how the tubules don't take actually a straight pathway. We discussed in the theoretical part how we have different regions of enamel that have different characteristics. The first layer we see near the enamel is called the mantle dentine, which is 20 to 150 micrometer in width. It is 5% less mineralized, and the collagen fibers in it is perpendicular to the dentino enamel junction. In this area, we see the branching of the terminals of the dentinal tubules, and it has a different mineralization process you will take in the dentinogenesis lecture. Another phenomena that we can see in dentine is what we call interglobular dentine. As I have mentioned, the dentine mineralization process starts as small calcipheres that grow to fuse together to form uniform calcified tissue. If these calcipheres fail to fuse, what happens is they stay as calcipheres and they look black in ground sections when they are seen under a transmitted light. A higher magnification can show us these calcipheres, and as you can see, the odontoblastic tubules pass through them without any problem. Also, the intertubular dentine is deposited there without any problems, as of course they are under calcified interglobular areas, so they don't disturb the pathway of these odontoblastic processes. Another slide that shows us some calcipheres that failed to fuse together and stayed as interglobular dentine. Another slide that shows us the calcipheres, the calcification centers of dentine that grow and grow until they fuse together to form the uniform calcified tissue. In the root, the odontoblastic processes at their terminal, they branch and branch, and actually they loop on each other. So when we make the sections, they are air-filled or stain-filled and show this layer of black dotted area near the cementum here. So this is the cementum. This is the hyaline layer we talked about in the lecture, and this is that dotted black area we called in the lecture granular layer of tome, which is caused by the looping of the terminal branches of the odontoblastic processes, that is air-filled or stain-filled. Also, it can be caused by some calcifiers that did not actually fuse together. This amorphous layer found between the cementum and the dentine is called the hairline layer. We don't know exactly if it's originated from the dentine or the cementum. It's around 20 micrometer in width. As you can see, it's atypical and structureless, and it helps bonding the dentine into the cementum. You can see here the odontoplastic processes and how they branch here to form granular layer of tome. In young teeth, sometimes we lack this hyaline layer. There is a direct contact between the cementum and the dentine. As you can see here, there is no hyaline layer in between. There is direct contact between the cementum and the dentine, while the branching happens here also and causes some kind of looping. At the other end, near the pulp, we found another kind of dentine that we called predentine. So this is the layer of odontoblasts that lies in the pulp tissue. They are the cells responsible for secreting dentine. The first layer of dentine, which is 10 to 40 micrometer in width, is called predentine. It's less mineralized than the circumpulpal dentine. As you can see, these are the calcifiers, the calcification centers that starts the calcification. As we progress toward the enamel in the dentine tissue, they fuse and make the uniformed calcified tissue. Another section that shows us the odontoblasts, the predentine, the calcification front, which is the name of the calcification centers that forms at the beginning of the mineralization, and here is the calcified dentine, and the tubules are very obvious in the tissue. Now we will talk about the structural lines that we can see in dentine. Some of them are related to the primary and secondary curves of the dentinal tubules, and some of them are incremental lines like the ones we have seen in the enamel. So one of the lines that is related to the primary curvature of the tubules is a Schrager line, which is the coincidence of the coincidence of the peaks of the S shape of the primary curvature of the dentinal tubules. When they coincide the peaks, they form this line that we call Schrager line that can be seen in longitudinal sections. Do you remember these lines? What did we call them in the enamel lecture? If you don't remember, go back and see what did we call these lines in the enamel. Right. As you can see in this slide, the periphery of the dentine near the enamel is called the mantle dentine. So of course, this is the dentino-enamel junction. The area under the mantle dentine into the pulp is the circumpulpal dentine, and this line shows us the primary curvature of the dentinal tubules. It is the coincidence of secondary curvatures of the dentinal tubules, which is hard to you now to recognize. It needs some experience to recognize the contour lines of Owen. So I would recommend you to see some slides online to get used to more to see Owen lines, contour lines of Owen. Again and again, this is the enamel, dentino-enamel junction. As you can see, it's not scalloped here, so most probably we are in the lateral surface of a tooth, whereas there is no much occlusal forces. This is the mantle dentine, the primary curvature of the odontoblastic tubules, and the circumpulpal dentine. Of course, here is the pulp. Another section here where we can see the enamel, the cementum, and the dentine. Which kind of relationship can you see here between the enamel and the cementum? The cementum is overlapping the enamel in this section. So these are the dentinal tubules, as you can see. Near the root, this area, this dark area between the dentine and the cementum, is called the granular layer of tone. Underneath it here, we can see a lot of calcipheres that failed to fuse in a phenomena we called interglobular dentine. This area is the mantle dentine, and underneath it starts the circumpulpal dentine. Of course, this is the dentino-enamel junction, and this is the dentino-cemental junction. Two types of incremental lines can be seen in the dentine: the von Ebner's lines and the Anderson lines. The von Ebner's lines are the lines that represents the daily rhythm of dentine formation. They lie perpendicularly on the odontoblastic tubules, and they can be seen only under high magnifications. A higher magnifications can show us these von Ebner's lines. The Anderson lines are the lines that represents almost weekly formation of dentine. So there is 6 to 10 von Ebner's lines between every two lines, Anderson lines. As we have discussed in the theoretical part, unital lines are exaggerated lines that are a kind of incremental lines in dentine and enamel. So neonatal line in enamel can be considered a line of stria, and a neonatal line in dentine can be considered one of the Anderson's lines. But both of them are exaggerated because the enamel and dentine that are formed before birth are different than the ones that are secreted after birth. Another line that is related to the secondary curvatures of odontoblastic tubules, which is one of the contour lines of Owen, is the line that happens when the primary dentine tubules change their direction in the secondary dentine. And as we have said, secondary dentine is a physiologically formed dentine that is formed after the tooth root formation is finished and the tooth is in occlusion. So we see this line. This is one of the contour lines of Owen between the primary and the secondary dentine. One also of the physiological phenomena that we can see in dentine with aging is the occlusion of the dentinal tubules, which makes the refractive index of the peritubular and the intertubular dentine match, which makes the dentine translucent. So we can see When we put it in water, they have the same refractive index. We can see what is behind the tooth. When we cut the tooth in cross-section, we can see the translucent dentine in a butterfly shape due to the convergence of the tubules from the enamel or from the surface into toward the pulp, where the surface area is much smaller. So this slide I showed you in the lecture, but the cavity wasn't included in the picture I showed you. So this tertiary dentine was formed because of the carious lesion and the cavity preparation. Both induced the pulpal cells to differentiate into odontoblast and form tertiary dentine, either to defend itself or to repair itself. So as you can see, the dentine the circumpulpal dentine is uniform in structure and in mineralization, while the tertiary dentine structure and form and size it's not as uniform as the primary dentine. Taking a higher magnification, you can see the difference in structure and in mineralization between the tertiary dentine and the primary dentine. Again, higher and higher magnification to show us that sometimes the tertiary dentine is amorphous, structureless. Also another slide that shows us how tertiary dentine is much different than primary dentine. So what happens if we have slow-growing stimuli like this fissure caries or like attrition? What happens is the tubules are occluded by dentine, which gives us this area that lacks structure and is transparent, and we call sclerotic dentine. If the stimulus is so fast or so harsh, the odontoblasts at the end of the odontoblastic tubules are dead, which leaves the odontoblastic tubules empty. So under section, they are air-filled and they look black. We call this phenomena the dead tracts of fish. Also, the dentine above the odontoblastic layer is most probably sclerotic. Another slide where we can see also a big carious lesion that has caused the death of the odontoblast. The cells here, the pulp cells tried to differentiate and secrete tertiary sclerotic dentine to protect the pulp. The tracts or the tubules are empty, they are air-filled, and they are called the dead tracts of fish. I will recommend strongly to you to search some slides on the website and try to recognize the structure yourself. I will be happy to help you. If you need any help, you can come to my office hours or send me an email with the slide that you can't read. And of course, that will help you to recognize these histological structures by yourself. I hope you enjoyed the lab. Wish you all the luck and bye bye.